Hi, thank you for listening to me speak today. I'm Lily Andrews, a PhD student at the University of Bristol, funded by Cancer Research UK. My talk is about the prevalence of BRAF V600 in primary gliomas, a systematic review. Brain tumours cause the most years of life lost by any cancer. This highlights the need for further research into the understanding of tumorigenesis in brain tumours. Due to the lack of treatment options in brain tumours, there is a need for a novel precision medicine approach which is informed by biomarker research. One potential candidate is BRAF, a gene responsible for the production of BRAF protein involved in the activation of the MAPK cell signaling pathway, leading to cellular processes such as growth and division. The most reported oncogenic driver mutation of BRAF is BRAF V600E, which is seen in around 90% of cases. Other V600 mutations exist, such as V600K and V600D, dependent on the amino acid substitution. Previous studies have identified varying prevalence rates of BRAF V600 mutation in glioma. For example, Breton et al. Found the BRAF V600E prevalence as 29.5% in their cohort. Conversely, Bailing et al. Found the prevalence to be as low as 1%. The primary objective of our study was to determine the average prevalence of BRAF V600 mutations across all diagnostic categories within the WHO classification and across all age groups within a clinical setting. Our protocol for the systematic review was registered on Prospero. A literature search was carried out on the 12th of October 2020 on Medline and Embase. Records were screened for eligibility and data extracted for records which were included. All included records were assessed using Hoy et al. Risk of bias tool. Meta-analysis was carried out on our prevalence estimates, and they were transformed by Freeman-Tukey double arc sign transformation and a random effects model. R was then used to create forest plots according to glioma WHO classification and age group. Our review consisted of 182 included studies with 13,669 glioma patients. On the right, you can see our prisma diagram to see the process of obtaining our studies. There was a large age range on our cohort ranging from 0 to 90 years. Majority of our studies had a low risk of bias, however, there were still some that were moderate and high risk of bias studies. Epithelioglioblastoma was found to have the highest average BRAF V600 mutation prevalence of 69%, with a 95% confidence interval of 45 to 89%. This result included 9 included studies, each with a square representing the prevalence of each study, and the line representing the 95% confidence interval. N is the number of patients with the mutation, total is the number of patients tested, and weight refers to the contribution of each study to the overall result. The I-squared value is the percentage of variability in the effect estimates due to heterogeneity. The tau represents among study variants. The dotted line represents the overall prevalence, and the triangle represents the 95% confidence interval. Pleomorphic xanthoastrocytoma was found to have an average BRAF V600 mutation prevalence of 56%, with a 95% confidence interval of 48 to 64%, with 36 included studies. Anaplastic pleomorphic xanthoastrocytoma have an average prevalence of 38%, 95% confidence interval of 23 to 54%, with 17 included studies. Ganglioglioma was found to have an average BRAF V600 mutation prevalence of 40%, with a 95% confidence interval of 33 to 47%, with 47 included studies. Anaplastic ganglioglioma has an average prevalence of 46%, with a 95% confidence interval of 18 to 76%, with 5 included studies. Here are the other glioma entities with BRAF V600 mutation prevalence of greater than 1%. Astroblastoma 24%, desmoplastic infantile astrocytoma at 16%, low-grade glioma not otherwise specified at 11%, subepidermal giant cell astrocytoma at 8%, glioneuronal tumour at 6%, dysembryoplastic neuroepithelial tumour at 3%, diffuse astrocytoma at 3%, pilocytic astrocytoma at 3%, glioma not otherwise specified at 2%, and polymorphous low-grade neuroepithelial tumour of the young had a prevalence of 50%. However, this result was from only one study with 6 patients. Glioma entities with a high prevalence may have similarities in their genetic landscape, but the relationship between these entities are poorly understood. One study has observed epithelioglioblastoma to have clinical similarities to anaplastic pleomorphic xanthoastrocytoma with epithelioid features. Another study has identified analogous features between epithelioglioblastoma and pleomorphic xanthoastrocytoma. Furthermore, case studies have identified epithelioid glioblastoma arising from pleomorphic xanthorastrocytoma. Over 99% of BRAF V600 mutations were found to be V600E in gliomas, but 2 were V600K, found in a pilocytic astrocytoma and a ganglioglioma. There was also one V600G mutation found in a ganglioglioma patient. However, many of these studies used immunohistochemistry in their analysis, which only identifies V600E mutations. Next-generation sequencing is needed to find the spectrum of mutations. To our knowledge, this is the largest systematic review and meta-analysis of BRAF V600 in glioma. Majority of our included studies had a low risk of bias. Our meta-analysis had a high I-squared value, however, this is normal for analysis of prevalence. There are varying techniques such as Sanger sequencing and immunohistochemistry to identify the V600 mutation between the studies included in our review. Diagnosing different glioma entities can also be difficult when there is discordant results between histology and genetic features. This can lead to misclassified glioma subtypes. My colleague Zach Thornton has looked at BRAF treatment response in V600 mutant glioma using RANA and RECIST imaging criteria. He looked at 394 V600 mutant gliomas from 130 publications. Complete response was in turquoise, partial response in blue, stable disease in orange, and progressive disease in purple. You can have a look at his poster for further details into his research. In conclusion, we present the largest systematic review of BRAF V600 looking at 182 studies and 13,669 glioma patients. Targeted testing is suggesting 5 glioma entities due to high BRAF V600 mutation prevalence. Glioma entities with a high BRAF V600 mutation prevalence alongside promising BRAF treatment response in V600 mutant gliomas provides rationale for adjuvant clinical trials of BRAF therapy. I'd like to thank all those who helped in this project who are listed here, and if you have any questions or comments, please feel free to email me, and thank you very much for listening to my talk today.